Okay, guys, what's up? We're gonna go through the Picornaviridae multiplication cycle handout. This would be a good review for me, and probably I hope to make these handouts make a bit more sense for some of you. So let's get started. So here, this right here is the virus, of course. And you can see it's written in here that it's made up of VP1, VP2, VP3 capsid proteins. And poliovirus will use the VP1 to attach to the receptor. So you can see here there's that VP1 right there. Okay. So the virus receptor is a VP1. The cell receptor is I think it's CD1, yeah, CD155 right there. So you can see that right there. So then when it attaches, then you have the partial uncoating. So partial uncoating means that it loses the capsid on the outside, but then there's still like a sort of like an inner capsid, like nucleocapsid, that's still left over, and you can see that right here. VPG, this VPG, think of that as something similar to a 5' cap for other RNA molecules, but for polio they use VPG instead of 5' cap, and I'll get into more detail later. But the reason why they use VPG is so that they can do have uridylation, so they can circularize the RNA and have it replicated. A sub n, that's just your polyadenylated sequence. A is for adenine, n is for, you know, n number of adenines that there are. So in here, I'm going to follow this one. You basically, this upper pathway is one way of seeing. I'm guessing some Picornaviridae, they sort of like inject their RNA into here. Others they go into the cell. So an example of polio, they actually go inside the whole partially uncoated virus goes into the cell. Inside of a vesicle, and once it's inside this vesicle, you have a pH drop within the vesicle, and that then allows the VPG to come, or the positive single-stranded RNA to come out into the cytoplasm. So transcription, replication, translation all takes place in the cytoplasm because it's an RNA molecule. It's not like influenza where it needs to steal 5' cap so it doesn't go to the nucleus. It's not a DNA virus, so it doesn't go to the nucleus to transcribe messenger RNAs and to replicate its genome. So this is all within the cytoplasm. So with either pathway, you have the RNA go into the cytoplasm. So you can see here, positive single-strand RNA. The first thing that polio does, or Picornaviruses in general, is they translate their genome, because if you recall from the beginning of the quarter, you can always think of Picornaviridae as a messenger RNA molecule. It can be immediately translated, and then it can be replicated after the initial translation. And this is why Picornaviridae is one of the fastest replicating, fastest multiplying viruses. So anyways, you have this basal level of translation. So you make a small amount of proteins. And you can see here, Dr. Samuels in this illustration, he's trying to show the open reading frame. So essentially this thing I just circled here, that's the open reading frame. There are three there are three, I guess, regions to the three open reading frames to the polyprotein precursor: P1, P2, P3. P2 and P3 code, no, P1 code for structural proteins. So it basically codes for your VP1, VP2, VP3, VP4. P2 and P3 code for non-structural proteins. So that's stuff like your 3D polymerase, your PR2A, which will degrade elongation initiation factor 4G. It encodes stuff like your 3C, your 3C protease, VPG, which is essentially the 5' cap that can be uridylated. So where's my mouse cursor? There we go. So essentially you translate this whole sequence at a basal level first. Now, Picornaviridae proteins, when they're being made, they have to go through a maturation step, just like any other proteins. And it does autoproteolysis. And from there, you get PR2A, PR3C. You know, you get your other proteins too, like your 3D polymerase right here, your VPG. Long story short, you now set up a base infrastructure for you to start making the negative single-stranded RNA from your one single strand of positive RNA. You can see that here. And with all the machinery put in place, you can now make a bunch of negative single-strand RNA. And of course, this is just a replicative intermediate. The negative single-stranded RNA's only function is to serve as templates to make a bunch more of positive single-stranded RNAs. Right? You can see that there's a bunch of positive single-stranded RNA here to make it as fast as possible. Right? So yeah, that's what the native ssRNA serves as. You can see there's percentages. So out of all the nucleic acids that polio will make in this multiplication cycle, 5% will be native single-stranded RNA, 95% will be positive single-stranded RNA. Okay, so up until now we have a bunch of positive single-stranded RNA replicated via the native single-stranded RNA intermediate, which itself was made from the original positive single-stranded RNA, plus all the base level of proteins, machinery, and so forth. Now that you have a bunch of positive, or essentially positive ssRNA, which is essentially mRNAs, you can now translate a bunch of proteins super fast too. And that is shown here. So you follow this red line that I'm drawing right now. That is shown there. So now you just make a bunch more proteins, even more PR2A, even more PR3C, even more polymerase and VPGs, and you can repeat the replication cycle and so forth until you deplete the cell's pool of nucleotides and so forth. But long story short is, with all the huge amount of positive single-stranded RNA, you can now make a bunch of proteins all at once. This arrow starting from VP031, that is just showing protein maturation. So VP031 is just your precursor, or like your immature VP1, VP2, VP3, VP4 proteins. You know, mature. Okay, now you have a bunch of structural proteins, a bunch of structural proteins, a bunch of positive ssRNA genomes. What you do, I said this a lot in the other previous videos, what do you do when you have all the structural proteins and a bunch of genome molecules? Well, you just assemble. Now it's time to get out. So you can see here, mature virion, and you have viral release. And with the viral release, polio kills the cell. Okay, so this is the Picornaviridae multiplication handout. I'm going to move on to the Rhabdoviridae handout now.